So Google quietly announced AlphaGenome, which is first of its kind to predict how single variants or mutations in human DNA sequences impact a wide range of biological processes in a comprehensive manner. It killed these benchmarks where it improved in 20 to 25% in some key metrics, and Professor Mark Mansour said, AlphaGenome will be a powerful tool for the field. Determining the relevance of different non-coding variants can be extremely challenging, particularly to do at scale. This tool will provide a crucial piece of the puzzle, allowing us to make better connections to understand diseases like cancer. So what is this? So this genome is our cellular instruction manual. It's the complete set of DNA which guides nearly every part of a living organism. Appearance and function to growth and reproduction. Small variations in a genome's DNA sequence can alter the organism's response to its environment or susceptibility to disease. But deciphering how the genome's instructions are read at the molecular level, so I'm not exactly sure what that means, but let's continue reading. Today we introduce AlphaGenome, a new AI tool that more comprehensively and accurately predicts how single variants or mutations in human DNA sequences impact a wide range of biological processes regulating genes. This was enabled, among other factors, by technical advances allowing the model to process long DNA sequences and output high-resolution predictions. And we'll read into this more on how this really helps with the disease. So basically, it takes a long DNA sequence as input up to 1 million letters, which is a pretty large amount, larger than other models, also known as base pairs, and predicts thousands of molecular properties characterizing its regulatory activity. It can also score the effects of genetic variants or mutations by comparing predictions of mutated sequences with unmutated ones. So in simple terms, that just means that the AlphaGenome reads long DNA code to predict how it works and how it changes and how it might affect the human body. So the most important use cases for this tool is disease understanding. So by more accurately predicting genetic disruptions, AlphaGenome could help researchers pinpoint the potential causes of disease more precisely and better interpret the functional impact of variants linked to certain traits, potentially uncovering new therapeutic targets. We think the model is especially suitable for studying rare variants with potential large effects, such as those causing rare Mendelian disorders. So synthetic biology, its predictions could be used to guide the design of synthetic DNA with specific regulatory function. For example, only activating a gene in nerve cells, but not in muscle cells. Fundamental research, I'm not going to go through that. For example, we used AlphaGenome to investigate the potential mechanism of cancer-associated mutation in an existing study of patients with T-cell acute leukemia. Researchers observed mutations at particular locations within the genome using AlphaGenome. We predicted that the mutations would activate a nearby gene called TAL1 by introducing a DNA binding motif which replicated the known disease mechanism and highlighted AlphaGenome's ability to link specific non-coding variants to disease genes. So yeah, that is pretty cool. I'm especially excited for this tool because cancer and heart disease are like the leading diseases in the US, and the sooner that we get this cured and prevented before that actually happens, the better. And it really looks like they improved the model capabilities from previous models by quite a lot. It's also available in the API if you want to use it as well.